So first, I'd like to apologize for getting here late. Thanks for American Airlines, and this one a result of a water skiing accident. But what I'd like to tell you about the importance of tumor heterogeneity and tumor evolution. Compared to normal cells, tumors are very highly heterogeneous, and there are many sources of this heterogeneity. Tumors have genetic alterations, so they have a genetic heterogeneity. Epigenetically, they are unstable, and also the microenvironment of the tumor is distorted that results in heterogeneity. And this leads to a very messed up transcriptional network in the tumors that basically enables them to change phenotypes and adapt to changes very easily. Why do we even care about this intratumor heterogeneity? As has been shown previously, and I'll show you later, it's a prognostic and predictive biomarker that can be used for patient stratification. It's also a tool to trace back tumor evolution, which is relevant for both the primary and secondary prevention, such as prevention of recurrence and the metastatic progression. It's also important for developing better experimental models of human cancer. The animal models that we have used traditionally don't reproduce the heterogeneity what we see in patients that could explain why their predictive value has been relatively poor over the years. And lastly, it's required for the design of optimal individualized therapy. So tumor evolution or tumorigenesis is a Darwinian evolutionary process, and therefore the principles of evolution, such as variability for hereditary phenotypic traits that impact fitness, reproduction, and selections are basically the drivers of evolution. Therefore, heterogeneity is really important in tumors. There have been a number of evolutionary models proposed in human tumors over the years as our techniques and approaches have changed. One of the first and simplest way of proposing a tumor evolution is this linear evolution, which basically suggests that there is a dominant clone that progressively accumulates mutations or changes, and that's what's really driving the tumor. Branching evolution proposes that there are actually subclones or that have different mutations, and then these can be have variable responses to treatment. Neutral evolution suggests that there is heterogeneity, but none of the clones really dominating and selected for. And lastly, this punctated evolution suggests that there is heterogeneity generated not in continuous way, but there are bursts of heterogeneity followed by more dominance of a particular subclone. And the heterogeneity in these different models usually increases as the tumors progress. The only one that proposes that there is a very low heterogeneity in tumors is the linear evolution, because that assumes there is always a dominant clone that takes over the tumor. But in all other models, actually there is an increased heterogeneity. And the data in the human tumors mostly reproduce the branching evolution and punctated evolution models in most tumors. How can we actually measure heterogeneity? Because if we want to use it as a biomarker, we need a very quantitative way of measuring heterogeneity. And for this Shannon index of diversity, which is actually borrowed from ecology, has been used commonly to assess quantitatively tumor heterogeneity. In nature, you would count how many different species and how many individuals in a species you see. Translating it to cancer, basically you can have a cancer cell with a unique identifiable feature as a species, and then you can assess how many of those are within a tumor, and also the topologic distribution of these cancer cells is also important. I'll just show you some examples of how we can actually visualize this heterogeneity in the tumors at the cellular level. These are assays where you can see that within a same breast cancer, different regions are very highly heterogeneous. The colors reflect cancer cells with different genetic alterations or phenotypic changes. And then as you see during treatment, these tumors change, and then you can very visually see this. This is another way of depicting, again, genetically heterogeneous cancer cells within tumors. And the heterogeneity in different regions and during treatment is very extensive. There is also a functional consequence of heterogeneity, because heterogeneous tumors behave differently than homogeneous tumors. there are actually interactions among subclones. There have been a number of studies suggesting that their cancer cells can directly communicate with each other and enhance each other's ability to grow, or they can modify the microenvironment, including the immune system, such as, for example, you can have a minor clone, a subpopulation of cancer cells that produces an immunosuppressive molecule that can favor the outgrowth of the whole tumor. We also seen that polyclonal tumors that have multiple subclones, they're more likely to metastatize, and they're also more likely to resist therapy. And all these data have suggested that cancer is really a systemic disease because these interactions among the subclones and in their microenvironment have systemic effects, including, most importantly, the influencing on the immune environment. From the clinical point of view, we also know that intratumor heterogeneity is very relevant, and this is getting incorporated into clinical practice more and more, particularly, for example, in breast cancer, where you can actually assess cellular heterogeneity. Multiple studies have shown that higher pretreatment diversity or heterogeneity is associated with therapeutic resistance. This is regardless of tumor type and regardless of treatment you're applying. This seems to be true that more heterogeneous tumors are more resistant to treatment. It's not just the number of different cancer cells, but also the topologic heterogeneity, like in solid tumors, seems to be prognostic. And basically what this suggests that we need to apply these tumor evolution models to design optimal cancer therapies and try to slow down evolution of the tumors instead of selecting for the most resistant and most aggressive subclones. And there have been a number of fairly limited approaches to apply this in the clinic. One of them is the adaptive therapy, which has been proposed by Robin Gettenby, and there is a clinical trial going on in prostate cancer testing anti-estrogen that instead of continuous dosing, you actually have a treatment vacation and alternate this depending on how the patient behaves, and that seems to be promising because it slows down expansion of resistant subclones. Another way to do it is to have drugs that actually would target the various mutations and the resistant subclones. That has been successful in EGFR mutant non-small cell lung cancer, where you have drugs that can target EGFR activating mutations and also this very common resistant subclone, the T790M mutation. And then lastly, as already mentioned by Charles, combinations are really the way to go to treat heterogeneous tumors, but designing rational combinations is not so easy, and running the clinical trials with those rational combinations are even more complicated. But in BRAF mutant melanoma, the BRAF MEK inhibitor therapy has been shown to be improve outcomes and decrease the expansion of resistant subclones. And hopefully we see more of these being incorporated in a clinic in the coming years. Thank you.